and opening up the webinar.
Okay, so your update is done? Tony?
Yes, we're starting.
No, your update was done?
Yes, all done.
Okay, great. Okay, thank you so much. I mean, without you, I mean, I cannot do anything.
You're welcome.
Okay, so we can chat, I mean, a little bit, and we normally wait a couple of minutes. So what's your plan for holidays?
I think my in-laws are gonna come. So my wife's sister and her mom and all the kids and grandkids are gonna be in right after Christmas.
Wow.
So hopefully do some fun things. Maybe we'll get some snow. There's nothing on the ground here right now. So
that's interesting. Wisconsin. I always, thinking Wisconsin always, covered with the snows.
Not in the last couple of years, yeah. Madison has dropped below the big snow line. So if you go about an hour and a half north of here, they got a big storm earlier in the week. And so, if you go up to the Twin Cities, I think we were talking to somebody up there the other day, and they have over almost 2 feet up there. So
I see. Yeah, we have the very weird tornado a few days ago. I mean, not a days ago. I mean, that's very weird because in the December, oh my God, I mean, I never seen this kind of big one. So.
Yeah, we were lucky that the storm that came through last night here had 60, 70 mile an hour winds, but we didn't see any tornadoes. It kind of dissipated as it got into the Madison area.
I see. Yeah, that's kind of very intimidating. I mean, because luckily we are in the city, so city was okay, but we have the Amazon warehouse nearby, and that is completely destroyed. I mean, as you saw, I mean, in the news. I mean, so how about Oregon?
That was crazy. If you saw on Saturday morning, the radar, the line of thunderstorms along the front stretched from the Gulf Coast all the way up into Ohio and Pennsylvania, unbroken. I mean, it was
right. Intense. Yeah, very, very intense, and that I would say really climate crisis right now. So we should do something. Otherwise, there is no future for my daughter, my younger generation, and all of us, unfortunately. So
synthetic biology, right? We got to save the day.
Yeah. And I hopeful because we have many young, young leaders like you. I mean, so I'm very hopeful. So.
I've been really surprised. Like it's been really hard to recruit in the last 2 years. And a lot of people I've had applied to my lab are basically kind of interested in jumping from a different industry. And they're very, very often are I want to work on something to push sustainable technologies forward. So
you're doing very cool science because you are from the Shuri's lab. I love him. and then you're doing the very, very interesting sequencing, technology. And I love it. I mean, and unfortunately, my colleague, I mean, just started the company, but he's passionate about company and, quit the UCLA. So that's already kind of sad, but at the same time, also kind of optimistic because, I mean, some technology, I mean, some people should do some technology development like him. So
yeah,
we'll see, I mean, how that goes.
Yeah.
So Brian, do you know the Shri? I mean, he's used to be the UCLA, now the start company. He's from, pardon?
I don't think I do.
Okay, so he used to be Georgia Church lab, I believe, right?
Yep.
And then he studied UCLA probably, I don't know, 7 years ago or 8 years ago. I don't remember.
I think 2014, if I remember correctly.
Right. And then he's super active, got a lot of grants from NIH. Probably, Brian, you probably do not know because he's doing more medical related work and also sequencing and also promoter kind of structure kind of characterization type of work with the Georgia Church before and then in his lab. But very super cool thing. I mean, we may hear something from Calin today.
Oh, nice.
Okay, I guess we need to start. Let's start. Okay, so this is the last of this year. So good morning, afternoon, evening, hola, hello, bonjour, ni hao, konnichiwa, 안녕하세요. Hello all. Welcome and thank you for joining us. The 14th seminar and BNC Day. And you'll see why I call it BNC Day. It is the last SynBYSS seminar this year, and then I can't wait the holiday break. I want to reflect on this seminar series because this is the last seminar of this year, but of course we'll come back in January. SynBYSS began on August 26th this year to help young people with exciting support from 80 pioneers, including a Nobel laureate. I'm talking about Francis Arnold, and 11 US National Academic members, and pioneers like Brian, today's speaker, and 88 rising stars, almost all the US government funders, many top journals, including Nature Journal and Cell journals, and over 1,000 global synthetic biology community members. The number of rising star speakers keep increasing, and they are working in many different regions. 71 from the United States, 9 Europe, 4 Canada, 3 Asia, and 1 United Arab Emirates. The pioneer speakers are from the 54 United States, 14 Europe, 4 Asia, 1 Australia, 1 Canada, and 4 Nature Journal editors, and 2 Cell Journal editors, and some other chief editor in many different journals. Obviously, more diverse people from different parts of the world should participate in this seminar series to make it a truly global event, although the time zone difference led to the uneven speaker distribution. In response to many requests for the recorded videos, especially from people in inconvenient time zones, all videos have been released with permission from all speakers. When I checked the YouTube channel last week, the total views for 12 videos were more than 11,000. That means 900 views per video on average. The number is not more important than the global excitement, and I'm truly thankful for all your excitement and support for this seminar series. This year means a lot to me, thanks to all of you. And thank you so much. This is my great honor to have all of you, and of course, today's speaker, Brian and Kelly. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Brian Pfleger. He is a professor at University of Wisconsin-Madison. Many people know him because he is an editor of Biotechnology and Bioengineering, asking for your help to review papers. So I know because he was trained by Jay Kisling, so he is my academic uncle. Ironically or interestingly, Austin, one of my best PhD students, had been trained by Brian when Austin was an undergraduate student. So I happened to adopt my academic cousin, and I'm advising him as my academic son. So isn't it horrible, right? So Brian, the virtual podium is all yours.
Thank you so much, Taesik. And Thank you for all your hard work on this. It's truly remarkable. The statistics you just ran off, that's such an impact, especially around the world. And bringing young people into this field is what's going to keep it growing. And so my message to everybody out there is that there's a lot to learn from what's come before us. And so when Teger asked me to just speak generally about my career and how I got started, one of the seminal memories that I have was probably for my first conference that I had ever presented as an independent researcher. It was a very big meeting, international meeting, lots of prominent people, many of the pioneers who are in this series were in the audience. And I got up and I was really excited, and I talked about our burgeoning lab and all the exciting things we had coming. At the time, it was our first fatty acid production studies in E. coli. And I was thrilled, and we were starting to work with the Great Lakes Bioenergy Research Center here at the university. And so I had interactions with lots of different types of people, different backgrounds. So life scientists, computer scientists, systems biologists. And I was really excited by the idea of applying different techniques, and particularly computational tools, to analyzing our cells. And I got up at the end of my speech and said, wow, I think this is the future for engineering biology. We need to make sure we're using computational models and tools of this nature. And very elegantly, one of our senior statesmen for the field came up, pulled me aside and said, Brian, we've been doing that for 3 decades. What are you talking about? Why is this the future? And so one of the things I want to pass on to all of the younger folks out there is oftentimes you'll find that you have amazing ideas, and you'll think that perhaps you're the first one to think of it. I do a quick literature search. I had the awkward experience of having a great idea in the lab from some data a student brought to me. And then I would go and find out that Alexander Steinbuchel, among many people, he's a researcher in Germany, for those who don't know, had come up with the same idea 7 years earlier, and I just hadn't seen the paper. And so the last thing I want to say about synthetic biology, and this is my soapbox perhaps on this, Madison is a very strong history in life science, and particularly at the intersection of life science and engineering, so the applied biotechnologies, food science, things of that nature. And so the history here dates back in synthetic biology to Václav Cybulski, who's a biochemist or oncologist. And in 1974, he wrote in a textbook that with the advent of recombinant DNA technology, we're entering the era of synthetic biology. And so his view on this was that akin to chemistry, when it went from a descriptive science to something where you could synthesize any system you like to study questions that perhaps were not accessible naturally, we were now entering that phase in biology, because we'd be able to design and build genetically modified systems and test questions that perhaps aren't possible in natural systems. And so while I think there's a lot of excitement in synthetic biology, it's certainly not a new field. I think it's an evolved field. And so that's my perspective on this. And so, if I'm leaving everybody with one small message is please learn from your predecessors. We're all stepping on the shoulders of what's come before us, and on the shoulders of giants. And Teger, I'm flattered that you consider me a pioneer. I think we're more contemporaries than anything else. So while we're technically, I guess I'm your uncle, I view you as a friend. And again, thank you so much for the opportunity to speak to this audience.
That's absolutely wonderful. I love it. So especially when you talk about your first talk international conference, I have the similar experience, because I always thankful to my advisor, my PhD advisor, Chris Prater at MIT. And she basically sent me to many international conference when I was even graduate student, and also SynBYSS, NSF Center at that time. I'm even talking in front of Jay Kisling and many big guys. And at the time I was so naive, and then I was not afraid somehow. But I have one interesting kind of memory. I was fortunate to speak in many big conferences when I was very young. I mean, as a junior faculty, but I was completely frozen when I talked in front of Frances Arnold after her keynote talk. And I actually literally frozen for 20 seconds. And actually, I don't remember what happened afterward. So my talk was
I had a recent experience just like that, Tasek. So again, maybe for the younger folks, we still get scared about certain talks. For me, I was giving a seminar at UC Riverside, and Harvey Blanch, you know, a pioneer in many respects, you know, the mentors for Francis Arnold and for many other pioneers in this field, happens to be affiliated with the department there and was going to be in attendance. And Harvey is one of the smartest people I've ever encountered. Right. And I knew he would unravel my talk with a question that because of a project that wasn't yet publicly released, he was going to unravel the whole story I was telling, and sure enough, he did. So that was the most nervous I've been at a talk in ages.
Yeah, Calin, we are not, kind of make you worry about, but I heard, the self-survey, number one fear of people is of course death. And second, the most one is actually presenting in a public, domain. So that's kind of natural. So don't be afraid, or don't be don't worry about things. So that's a natural thing. So okay. So anyway, so of course, when I talked to her, during the seminar series, the first one, I was not afraid anything because I was just relaxed, and then she was far from me. Even the virtually. So I didn't bother at that time. So anyway, so great. So okay, now the main speaker of today with a longer introduction. Calin Plesa is an assistant professor at the University of Oregon. He received a bachelor degree in engineering physics from Simon Fraser University, and master in nanoscience from Chalmers University of Technology, and a PhD from Delft University of Technology. So that means, he's from Europe. As an HFSP fellow, this is one of the prestigious, very highly regarded fellowship. In the Kosuri lab at UCLA, he developed dropsin, a no-cost scalable method to synthesize thousands of genes. And Calin holds a Casey Award from the Boros Wellcome Fund. Actually, this private foundation is one of the SynBYSS seminar series speaker, and then started his lab at the University of Oregon 2019, 2 years ago. And his lab focuses on accelerating the pace at which we understand and engineer biological protein-based systems. Toward this end, of course, his lab also developed new technology for gene synthesis, multiple functional analysis, in vivo mutagenesis, and genotype-phenotype linkages for several different research areas and application. These allow both access to the huge sequence diversity present in natural systems, as well as testing of rationally designed hypotheses encoded onto DNA at much larger scales than previously possible. I believe this technology is the reason synthetic biology exists, because without this type of synthesis and sequencing technology, our current possibility or future probably very limited. So this is my great pleasure to introduce him today, and then of course virtual floor is all yours from now on. Thank you so much, both of you. This is such an honor to have both of you today.
Thank you for that introduction, Professor Moon, and thank you for organizing this series. It's been really good. So I think there was one particular sentence that Brian mentioned, which is kind of very relevant, which was, as soon as we were able to start actually synthesizing DNA, we could access and we could build designs and models that didn't necessarily exist in nature. And so that's kind of the key thing I want to kind of talk today about, and really kind of the key idea that we've been pushing forward as much as possible in my own lab. So mainly the talk today will be focused around two technologies. One is this drop synth multiplex gene synthesis technology that I developed in the Kosuri lab at UCLA. And then a second one is broad mutational scanning, which is kind of a way to look at sequence function relationships among a broad set of related proteins. So jumping in, of course, we all know that nature is extremely diverse, both at the macro scale and at the micro scale. We have this huge number of huge variety of different protein structures that we see. And if we look at databases such as UniPro, we can see that with the advent of next generation sequencing, we had this huge increase shown here on a log scale, where we now have many hundreds of millions of different proteins, and which is shown by the orange curve. And so unfortunately, we still don't have a very good understanding of what many of these proteins do. So the blue curve kind of shows some idea of how many of these proteins do we have some sort of experimental data on, or experimental characterization. Computational techniques, especially more recently, have made a big improvement in this area, but there's still many cases where a single point mutation can completely alter the functionality of a protein. For example, in transporters or in receptors, right? They can completely change the substrate or the binding ligand. And so we need new technologies in order to close this huge gap, which I think this is probably out of date now, but currently stands at over 125 million proteins. And so even within groups of proteins that supposedly do the same function, so what's shown here is the percentage identity between orthologs in the same group. So all families of proteins that supposedly carry out the same functionality. And even those proteins, we see extreme diversity at the sequence level, right? So you can see the mode of that distribution of around 40% or so. And so in addition to all this diversity that's out there in nature, we also have extreme complexity, right? I think this is a system from a photoreceptor complex. So tackling this complexity really kind of requires often many hypotheses, many designs, many shots on goal to be taken. And so, I got hired here at the Phil and Penny Knight campus for accelerating scientific impact at the University of Oregon. And, very nicely, we have this word accelerating right in our name. And so I think, my lab was a very good fit for that, particularly because the one thing that all of the projects in my lab have in common is this idea of tackling diversity and complexity by building technologies that can scale to many hundreds of thousands, many millions of hypotheses or designs. And so the key thing here is we generally focus on protein, and we want to move away from studying individual proteins, as was often done classically in the past, towards studying either entire protein families, be they functionally related, or even entire full proteomes where they're non-functionally related. And so how do we do that, right? Typically when I mentioned that, we work on scaling technologies, people are like, oh, you must have, a lot of robots. A warehouse of robots. I don't have space to fit a warehouse of robots. We don't really have any robots. But we do want to get away from this idea of having a single test tube test one design or test one hypothesis at a time towards something that's a lot more multiplex in nature. And the way we do that is something akin to imagine, going to a store, you put all these items into your shopping cart. Each of these items has a barcode. You go at the cash out counter, and the cashier will use the laser scanner to basically scan all the barcodes, right? So we effectively do something similar, except in our case, the shopping cart is a test tube, and all the items are essentially cells or molecules. And instead of using a barcode with a laser scanner, we have a barcode that's a small little piece of DNA with some usually randomly encoded sequence. And we scan those with a DNA sequencer, right? And the huge advantage is that, within the last decade or so, it's become extremely cheap due to the exponentially decreasing cost of DNA sequencing. So we can now, pay $1 and read, hundreds of thousands of different barcodes, which allows us to really do these assays at scales that were previously unimaginable. And so this is the idea, right? We're moving from individual test tubes to everything kind of mixed together and multiplexed in a single test tube. And so if we were to zoom inside this test tube, we would see one cell with some particular protein variant or gene variant attached to one unique barcode, and another cell containing another variant, which is attached to a different barcode. And we then go through two different sequencing steps, one a mapping step, where we link what is the barcode attached to each variant. And then subsequently in the assay, we only need to read out the smaller barcodes in order to determine how the individual variants have propagated through our population. So the way this typically goes is that we synthesize a very large library of genes, we attach them to barcode, we put them through these functional assays, and this can be any one of a number of things. We prefer very often a life-death selection. So we link the functionality of the particular protein or gene to the survival of the cell, such that any cells containing proteins that are functional get enriched, and so the barcodes also get enriched, and any ones that are not functional get depleted. But it is also, of course, possible to link the functionality to other things such as fluorescence, which can be sorted on a fax, or buoyancy, where you can just essentially scoop everything off the top of the tube. So the key question here and the key input to this pipeline is the ability to generate some gene library. And so what are the different technologies to actually make a gene library? So if you think about a plot where we're thinking about the diversity that we can access within this library and the maximum sequence length, we have a couple of different options. So the first option is shown at the bottom is just mutagenesis. We can take our gene and just mutagenize it. We can access that in any length, but unfortunately, it only allows us to access very low distance mutations from our original parental sequence. The second technology, which is shown on the left and in this video from Agilent, is microarray-derived oligos. So several decades ago already now, we came up with this technology, which was essentially something akin to an inkjet printer, except instead of printing different colors, we would print the chemicals, the phosphoramidites necessary to assemble an oligo. And you can do this on a glass slide, and you can print tens to even hundreds of thousands of different oligos. And the advantage that this gives is that the price or the cost per oligo is extremely low, on the order of several cents, maybe 10 cents, depending on the length. And so these oligos are extremely low cost, but they come with several different disadvantages. One is that currently the maximum oligo synthesis length is about 300 base pairs. You can go longer, but they become increasingly there's more errors in them. Secondly, compared to traditional column synthesized oligos, they do have quite a few more errors, in particular deletion errors, which of course for proteins can lead to frame shifts. And the final thing is that at the end of this synthesis process, everything just gets cleaved from the surface, and you get a single tube with all of your 100,000 oligos mixed together. So you need some sort of way to manipulate them in multiplex. Another approach, of course, is just PCR. The problem with PCR is you generally require the source. So if you're interested in working with a protein from, I don't know, some organism under the ice in Antarctica, it's going to be difficult to get. Generally, it's hard to design hypotheses, and it doesn't scale well. So gene synthesis is kind of an umbrella term for a whole bunch of different methods that take these smaller oligos or smaller pieces of DNA and assemble them into longer lengths, longer gene length fragments. Unfortunately, the primary drawback of gene synthesis is that it's got a high cost per variant. And so, if you want to do an assay where you're studying 5,000 or 10,000 or more genes, you're not going to be able to do that at the moment unless you have an extremely large budget. And so I started working on this technology we call DropSynth in my postdoc. And the idea here is to take these extremely low cost oligos and assemble them together in a multiplex manner, in a pooled manner, and while maintaining an extremely low cost per variant and pushing up towards lengths of 1,000 base pairs. So when we started this in 2015, the difference between the cost per base to read one base of DNA, which is shown by the orange curve, and the cost to write one base of DNA was approximately 1.3 million times. And after we went through multiple iterations of this technology, we were able to reduce that by several orders of magnitude to where the difference is only about 68,000 times. And so how does this work? We start off with a gene library in a computer, all the sequences together, bioinformatically designed or mined from some metagenomic database. We then, for each gene, split that corresponding gene up into multiple fragments, such that each fragment has a length that can fit on one of these oligos, so is under 300 base pairs. And then you might think, okay, so we received this pool of all these fragments, we can go in and just assemble them with something like PCA. And the problem is that due to cross-hybridization reactions, you can't do this. So the key kind of challenge here is that you have to be able to take the oligos from this mixture, this huge mixture of hundreds of thousands of oligos, and isolate only the fragments necessary to assemble your particular gene. And you have to do this in some way that's scalable and cost-effective and multiplexable. And the way we achieve this is by putting all of these fragments on an oligo and tagging them all with a gene-specific, what we call microbead barcode. So each of the fragments necessary to assemble one gene contains the same microbead barcode. And this is different from the microbead barcodes on other genes present in the library. After we synthesize this, we receive it from a supplier, what we do is we use type 2S nicking enzymes to expose the microbead barcode as a single-stranded overhang. We remove the smaller fragment, we melt it away. And we do this in parallel for huge numbers of oligos simultaneously. We then mix these with beads that display only one type of complementary barcode on their surface in such a way that each bead hybridizes only the fragments necessary to assemble some particular gene. We wash away everything that was not hybridized, and each of these beads is then processed in a simple vortex-based emulsion into a droplet. So in the ideal case, we have a droplet with a single bead inside, they're Poisson loaded, and we use a type 2S restriction enzyme to basically cleave the payload from the surface of the bead, releasing these payloads into the bead. And then we use a polymerase cycling assembly to assemble the full length gene inside the droplet. And what's key here is that this is done in millions of droplets in our emulsion simultaneously. So we're able to assemble many genes simultaneously. At the end, we break the emulsion and we recover our fully assembled gene library before tagging it with a random barcode, like so. And so what's key to understand here is that this entire process essentially relies on isolating each of these fragments from this large oligo pool by hybridizing them onto beads. And we can make many beads with each one containing different barcodes and do this in a highly parallelizable manner. And so when we released the first version of this technology in 2018, what you see here, each of these points represents a library of 384 genes. So we assembled 9,984 genes in a span of about 2 weeks. Of those, about 6,200 were successful. So we typically have a coverage of around 70% for each library. And that's the amount of genes within each library that after sequencing, we were able to see at least one correctly assembled sequence. We then carried out a number of different optimizations, looking at what polymerase to use, increasing the scale, implementing error correction, and I'll talk a little bit about each of these. So when we first started the initial version of DropSynth, we used kappa robust as a way to do PCA inside the droplets, and we had about 4% of the molecules actually being perfect. And so what I mean by that is, if we were to take one droplet corresponding to one gene out of these emulsions and break it open and look at the molecules inside. For every 100 DNA molecules inside that droplet, typically 4 of those would have the correct amino acid sequence. And so merely by switching over to a high fidelity polymerase, we're able to increase that substantially up to typically we see in the range of 20 to 30% perfects these days. And so although we only have, in the initial version of the technology, 4% perfects, the key thing here is that if you feed these into multiplex functional assays, they're able to oversample the library so much that even with only 4% and even actually less, we're actually able to get more than enough functional data on each of the library members present inside. And so with 24%, this allows us to actually access even longer lengths. And so one of the grad students in my lab now, Natalia Viegas, she's working on pushing this up using a number of different approaches, even higher. And the key reason why we want to do here is essentially because we want to go to longer lengths, right? So up to now, we've done around 800 base pairs, maybe a little bit longer as well. But we really want to get this in the short term up to about 1.5 kb. And the issue here is that the longer gene you want to make, the more oligos you have to use. And so the inherent errors present in those oligos as you have to attach more and more together get propagated through the assembly process. And so your corresponding percentage perfects after assembly continues to drop based on the errors present in your source oligos. And so, as I mentioned, short term, we're interested in getting this up to about 1.5 kb, and we kind of have a roadmap if that pans out to get it to much longer lengths after that. And so the coverage, what we're able to do is that we're able to maintain it about 70%, but by combining, essentially synthesizing 2 different codon versions for each protein we want to synthesize. And so essentially 2 different codon optimizations that are relatively distant from each other. And combining the 2, we're able to push up our coverage actually quite a bit to typically about 80% level without much additional work. And so the idea here is that there's particular sequence features in a sequence at the nucleotide level, which cause particular assemblies to drop out or be lowly represented or not assemble at all. And so the other thing we did in the second version was we increased the scale of each reaction, so each tube, from 384 genes to 1536 genes. And so we're able to do this without any detectable loss of the yield of the percentage perfects after assembly or any hit to our coverage. And so we're looking at different methods now to, over the short term, increase this to 6,000 or 12,000 genes per reaction, and over the long term, even quite a bit more than that. And so the other major interesting factor that was optimized was the cost. So if you look at the cost per kilobase pairs, exclusive of labor and equipment, at the 384 plex level in the initial version, it was about $1.20. And by scaling this to 1536, it basically went down to about 70 cents per kilobase pair. And so one thing I want to point out here is that the key factor dominating the cost of the technique right now is actually the cost of your underlying oligos. So as the cost of oligos will hopefully decrease in the future, we'll be able to piggyback on those trends and essentially get a corresponding cost reduction after assembly here as well. And so when we typically do drop synth, and so you run this PCA in the emulsion, you often see a wide variety of these low molecular weight products when you run the products on a gel, as shown here on the left side. We're able to integrate suppression Pcr effect, and so in this case, we use an inverted terminal repeat in such a way that the ends interact with each other and suppress priming based on length. So this effect is much stronger for shorter molecules than for longer molecules. And so this kind of selectivity allows us to automatically select for the higher molecular weight products as shown kind of in the middle lane that figure. So, you know, if I have to say what we do in our lab, we start essentially at the top of this diagram. We take sequences from metagenomic database, genomic databases, or from computational design tools. We use cheap gene synthesis to synthesize these all. We can synthesize tens of thousands of genes in a week at lengths of, you know, under 1 kb right now, very low cost, pretty fast. We then plug these into multiplex assays, which are able to test hundreds of thousands of variants simultaneously. We then sequence the results and interpret the results and feed those back into our pipeline. And so what's key here is that we've kind of designed this in such a way that we can piggyback on exponential trends in cost of DNA synthesis as well as cost of DNA sequencing, which we'll able to take advantage of as these costs become lower and lower. So I'll kind of briefly now give proof of concept for what you can do with this type of approach in terms of the characterization of a protein family. In this case, this enzyme here called PPAT, which catalyzes the second to last step in the conversion of vitamin B5 to coenzyme A, right? So coenzyme A is an essential cofactor present in all organisms. And what's interesting about this enzyme PPAT is that it has relatively low homology to its mammalian counterparts, the bacterial ones that is. And so it's been an interesting potential target for antibiotic development. And so what we went and did was we grabbed 1,152 sequences out of NCBI. We went and we synthesized those. Here you're showing the sequence level identity relative to E. coli, which is on the far right of this plot. We synthesized those all, and we created a strain whereby we knocked out the endogenous gene, PPAT, and we put it on a heat-curable rescue plasmid. We then took our synthesized library of all these PPAT homologs, and we plugged it into these strains. We exposed these to 42 degrees heat curing, removing the native gene on the rescue plasmid. And then we essentially allowed the cells to grow up. And so in this case, the entire metabolic system within each of these cells was dependent on the functionality of the PPAT variant that was present inside them, which we could keep track by the associated barcodes. And so some of these strains were enriched, some were maintained at a stable level, and some were depleted. And so I'll talk about these so-called fitness scores in the next slide. And so all you need to know about these fitness scores is essentially they're a measure of the abundance of each variant over time. So if you have a positive fitness score of 1, it doubled. If you have a negative fitness score of -1, it basically halved in population. And if you're 0, you maintained a stable level. So we ran this experiment. Here is shown 2 different biological replicates with very good correlation in between the 2. The negative controls, which were missing a key catalytic motif in the enzyme, showed very strong dropout of the population. And so here's kind of our phylogenetic tree showing, I think, 450 different homologs, and I haven't projected the fitness on here, but what we were expecting going into this is that, you know, a lot of the enzymes that were very relatively closely evolutionarily related to E. coli, which is kind of in the top right there, would do very well. Whereas some of the more distant homologs, and in some cases we had some extremely distant homologs taken from, I think, plants and eukaryotes, would drop out of the population really quickly. So that's not what we saw at all, as is often the case in science. And so I want to point out 2 key features here. And so what I'm showing here is essentially a color-coded view of the fitness, with the very bright red and orange colors being very strong fitness, whereas the dark black being extremely low fitness, essentially dropping out of the population. So 2 things were surprising. One was that 70% of all the homologs we tested were able to rescue function just fine. And secondly, the ones which did drop out did not cluster into clades almost at all. right? So there's kind of, as you can see, randomly distributed throughout this phylogenetic tree. And so why was that? And so in order to kind of answer this question, we developed this new technology, new approach called broad mutational scanning. And so what we're doing here is that imagine we have some particular function. In this case, PPAD, right? So you have many different regions of sequence space which potentially encode for this function. If we then zoom in on this functional space, we can determine the region explored by evolution as shown in green, and then a subset of that which we've seen through next generation metagenomic sequencing. And so what we did through this assay was we synthesized a whole bunch of different points throughout this sequence space. And because our synthesis process was essentially so bad in the initial version of this dropsum technology, as you saw, we only got 4% perfects. We got a lot of mutants for each design, which were relatively low distance away from each of these. And so what this allowed us to do was create a very low, shallow mutational scan around each of the corresponding design points. So if we look at the distribution of mutants inside, you see this kind of pattern here. And so at the top, we see all these mutants, which are very far distance away from the amino acid sequence we designed. Approximately 53% are very large frame shifts, primarily due to deletions. But if we look at the bottom of this, and not including the perfects, about a third of the sequences we found after mapping were relatively low distance mutants away from our design sequence. And so what this allows us to do, as I mentioned, is essentially do a low, shallow mutational scan around each of our parental design points. And so we got all this data together. So we built these heat maps up corresponding to the fitness of every amino acid at every position on every homolog. So we have 3 dimensions here. And we split this up into 2 things, a loss of function matrix and a gain of function matrix. And so for the loss of function matrix, we take only high fitness homologs. We then collapse it among all homologs, and we build this up column by column, row by row into a heat map that looks like this. And so what you're seeing here is essentially for each of these points, the median fitness of that amino acid at that aligned position in the context of many, many, many different backgrounds. So for many, many different homologs. And there's 2 things I want to point out here. One is that, areas, for example, that TXGH motif, which is shown on the left, which are critical to function, generally are not tolerant for any kind of mutations. On the other hand, though, areas where, at least in the E. coli version, are known to interact with the substrate, we see relatively high levels of promiscuity. So many mutations are actually permissible in those regions. And so that's what's shown kind of in this region, in this structure, where we've projected the average fitness for every residue onto the E. coli structure. And you can see that there's relatively low tolerance in the catalytic pocket, whereas there's relatively high levels of mutational tolerance effectively everywhere else. And so the other part of this was this gain of function matrix. And so what we did for these is that we took all of the homologs that were basically were dropped out of the population, right? So the homologs which were not able to complement in E. coli. We then did something similar where we collapsed among all homologs, and we collapsed among all amino acids, and we projected that onto this type of a plot. So what you're seeing here is essentially the number of gain of function mutants, right? So we selected specifically mutants from parental sequences which dropped out in such a way that those mutants basically restored function. And if you clustered these all together after collapsing those 2 dimensions, what you can see is that they tend to occur in specific positions. and if we project it onto the surface of the E. coli version, you can see that we can be relatively confident in this because they tend to cluster together, even in this case, for example, on two distant beta strands on a beta sheet. And so this data corresponding, taken together with a number of other related experiments that we ran, kind of pointed to this idea that the two key factors affecting dropout were actually, one, interactions with other cytosolic components. And so what I mean by that is evolution has basically has crafted and shaped the particular interactions that this enzyme can make in its own host. When we do heterologous expression and we take it from the native host and put it in E. coli, suddenly, it has all these new potential interacting partners, which have not been evolutionarily screened against. And so we believe that is one of the main effects causing these dropouts, together with some gene dosage toxicity effects related to the expression level that we used. And so that's kind of the initial proof of concept that we did on that. And so I want to briefly wrap up with mentioning a couple of different things we're working on now in the lab. One thing, as shown on the very far left, we're working on is looking at different ways to explore sequence function landscapes. And we're doing that using fluorescent proteins as a model system. We've synthesized essentially all known fluorescent proteins, and we're using that as a test bed for looking at different ways to explore protein functional landscapes and perhaps integrate machine learning models into protein engineering of that family. We're then also very interested in antibodies. So we're working on developing a platform to really be able to generate antibodies against many, many antigens simultaneously by leveraging these technologies that I mentioned today. And finally, we're also very interested in biosensors. So in particular, using receptor histidine kinases, which are essentially one of the largest protein families out there. There's millions of receptor histidine kinases that can detect pretty much any stimulus or ligand that you can think of. So we're also building some platforms to try to engineer and characterize these at very large scales. And so with that, I'd like to thank all the people in my lab and the people in my postdoc lab, which were involved in this work, as well as all of the funders which supported this work, which are shown on the right. And so we're recruiting. Come talk to me if you're interested in this type of technology. We've got lots of projects, lots of openings. I'd like to thank you for your attention and happy to take any questions you might have.
Amazing, amazing. Wow, fantastic. So I guess, young people should apply.
Hopefully.
Yeah, for his position. That's wonderful in the technology. I love it. So let me check a question. I actually do not see question now. That means your talk is perfect. So I'll start actually two questions I have. So this is the wonderful. So, but my question, that's question always I have. So to me, synthesis is not a bottleneck anymore in terms of cost, but validation by sequencing and QC to make sure correct sequences might be the limitation. I might be wrong, but so I want to hear your answer for that question. I'll ask the second question afterward.
Yeah, often, I think I often will think about these two together, right? So you can't really have the first one without, it's not as useful to just have a whole bunch of assembled sequences if you don't have the corresponding knowledge of, how many are good, which ones are good. And so they kind of really go together pretty strongly. I generally agree to some level. I think for some of the work that's being done, definitely synthesis is no longer a bottleneck. That being said, there's definitely a subset of assays and applications where we really want to go to much, much larger scales. And so, I've had people come and ask me like, I want to synthesize 1 million genes. Mm-hmm. And I think there's, there's demand for that. I mean, I don't think it's very strong right now, but I think people are starting to realize that we're kind of on the verge of that being possible. And when that becomes possible, there's a whole bunch of applications and studies that become accessible, which, you know, were definitely not even imaginable a couple of years ago. And so I think I would personally be happy if I could order, you know, a gene of, you know, however many kilobases for a couple cents, then I would stop working on gene synthesis, I think.
Okay, that's great. So actually, my second question already related to that answer. So my second question is when or what price would be the time or price I should consider all my student order all the genes completely stop doing cloning. So for library synthesis, a DNA synthesis is definitely cost effective or cheap, much cheaper. But I am curious about routine cloning. So for example, I heard many different things like if you fail to clone after a few trials and sequencing, synthesis would be better in terms of cost. And let's say I'm cloning 10 variants of plasmid with 10 kb gene each. And should I say, you know, to my student, order the 10 gene, or I still say, I mean, you should try cloning and sequencing in the lab. So what would be your answer? So what is the point?
I can tell you what we do in my lab is generally, so usually I have people do cloning just so they learn and they know how to do it.
Right.
Because I feel like it's a dying art because nobody does it anymore.
Right.
But that being said, usually I'll tell my students to go and order it from one of the vendors. You know, there's the cost of synthesizing genes has just reduced so much that if you think about the time it takes them to actually do the cloning, characterize it, you know, troubleshoot everything. Generally, I find that if you do the cost benefit comparison, it's worthwhile in order to free up a lot of time to just order it. So we order almost all of our genes, I would say, these days.
That's interesting. So the reason I'm asking that question is if we order, okay, let's say 1 kb gene, and then 10 of them, that might be cheaper. But if we're talking about same construct, but we are talking, well, still if we order the 10 gene, but still we need to clone it because into the vector. Mm-hmm. And then if we're talking about the total plasmid synthesis, that, you know, minimum 3 kb or 4 kb, I don't know that part, you know, that point, and probably the current synthesis price probably too expensive to do everything. Mm-hmm. So at the end of the day, you know, we need to do some things, you know, in terms of cloning. I love cloning because I generate, you know, more than 20 clone just one week. I mean, without any problem. If my sequencing verification fail, I'm very angry because I'm very good at it. But, you know, that's not necessarily true to, you know, many of the students, especially the beginners. Mm-hmm. And I'm still wondering whether ordering is cheaper or, so what is the price point? Let's say, so I'm not talking about library. Library we need to go with the synthesis. But in terms of cloning, let's say I'm talking about 1 kb, what is the price point I need to just order without just thinking about cloning at all?
I mean, if I think about, you know, the current price relative to, you know, what the price has been like in the past, I think it's already, even if you have some of the vendors do the cloning for you with that additional cost, I think it's still quite competitive at the moment. It depends. If you're really skilled, I guess it depends how much you value your time, right?
Right. If you consider time, yes, definitely we need to go to that. Yeah. And then, but at the same time, you also mentioned the very important point, the training. So the cloning, during the cloning, they can learn all the PCR concept and everything. Yeah. And
the other thing I also want to say is that, even if you order it fails still at a surprisingly high amount. We've had, I can think of at least 3 examples in the past year where we've had synthesis, complete synthesis failures.
Right, we did. I mean, we have the high GC the gene, because of my bacteria, one of my bacteria high GC.
Yep.
They miserably failed multiple times, and they said, "Oh, you don't need to pay anything." I mean, so
it's been we've had a whole bunch of failures for things that we couldn't figure out exactly. It wasn't GC content, it wasn't repeats. We think it might be some kind of cryptic promoters combined with some potentially toxic stuff in there, but it's still really tricky, because I feel like if you tackle that by yourself in a lab, you can get stuck pretty quickly on the troubleshooting. Whereas I think, a lot of these vendors have a lot of expertise and know exactly, how to modify the sequences and how to do things to actually get it working.
Okay, so
I think that is one of the major advantages of the library-based approaches, which is that you don't care if whatever fail, right? Because you still got and others that work.
Yeah, library we need to in order. I mean, definitely no question. But I still wondering which one is better. So I probably need to consider.
I feel like it's right now it's on the edge. For many people, it's still a little bit too expensive. I think if it could just get a little bit cheaper, maybe on the level of probably under 5 cents or under 4 cents per base. I think the adoption would be a lot stronger.
5 cents per base. Okay.
Yeah, something on the order. I mean, ideally about a cent per base, I would say, but I don't know.
Yeah, because I mean, when I synthesized for the first time, the cost was for 1 kb gene. The cost was almost $1,500 long time ago. So when I was a graduate student, that's a 2006 or something like that. So now, tremendously improved, but
so I think now, you're on the level of what, about $70 to $100 per 1 kb. So, I think it's improved massively. I think if we can get that down to, $10, $20 per gene, that would be really like, cloning would basically die off.
just and then I'm become the basically the remnant of the old. Okay, I guess we probably close up. So thank all for joining and staying today again, and then also thank you. That brilliant talk again. So we will meet again on January 6th next year after 3 weeks of break. And given the difficulty we all have been through, we all deserve a nice holiday break. And you do not want to miss the next seminar, however, because we will have the Terry Shepard, Nature ChemBio chief editor, and Alison Warner from National Renewable Energy Lab. And what else would be better than starting the new year with a great talk given by the top journal editor and a rising star. So as usual, the follow-up Zoom chat will occur via the link in the chat box, and see you in a minute. And for you, you can just click that link, and then that will ask whether you will go to another room, and then you can just say yes, and then you will move to another room, basically.
Okay.
Okay, see you in a minute. So Tony, thank you so much for entire year. Without you, I cannot do anything. And then I'm so happy to work with you entire, half year.
You're very welcome, Dr. Moon.
Yeah. So I'm looking forward to the next year, continue to work with you. And then, please send the recording to me, and then, and then happy holiday.
All right, happy holidays.
Yeah, so we deserve happy holiday, right?
Right. Take care now.
Enjoy your all the grandkids, grandchildren.
Yes, I'll have more fun with the grandchildren. Have a great time with your family.
Yeah, you too. See you next year.
Take care now. Bye-bye.
Yeah, thank you.